My dear students, welcome to BrainOscientia, your own classroom. This is the second part of the question series of Neet Biology for the chapter Cell Cycle and Cell Division. The question is that during which phase of the cell cycle the amount of DNA in a cell remains at 4C level if the initial amount is denoted at 2C. That means in which phase of the cell cycle the amount of DNA got double. That means initially the amount is 2C, now the amount is 4C. That means which phase of the cell cycle that is going to happen. So you see, cell cycle comprises of interphase and mitosis. Interphase again comprises of three phases G1, S, G2. So in the S phase of the cell cycle, chromosome got double, that is DNA got double. As a result, just after S phase, G2 phase starts, and where we'll get the double amount of DNA, because in the S phase DNA replication takes place. As a result, the DNA got doubled, and just after S phase G2 phase is there. As a result, in G2 phase you will find the DNA got double. The answer is only G. Next question, in S phase of the cell cycle the amount of DNA doubles in each cell, the amount of DNA remains same in each cell, chromosome number is increased, the amount of DNA is reduced to half in each cell. So in S phase of the cell cycle, what happens? The DNA replication takes place. As a result, the DNA got double. So answer is in S phase of the cell cycle, the DNA contents gets double, that is from 2C to 4C for diploid cell, because in S phase the replication of DNA takes place. This question has come in Neet 2019. The enzyme recombinase is required at which stage of meiosis? So we know meiosis comprises of meiosis 1 and meiosis 2. In meiosis 1 there are again prophase 1, metaphase 1, telophase 1, anaphase 1 and telophase 1. Prophase 1 is further divided into some phase steps, and the question is that in which stage of meiosis the enzyme recombinase is required. So in meiosis the sister chromatids, homologous sister chromatids come close together, and as a result chiasma formation takes place. This leads to the crossing over of this takes place. As a result, genetic recombination happens. In which place this recombination takes place? The answer is pechidin. In pechidin, recombination of genetic material, there is a chromosome takes place, and the enzyme recombinases activate at the pechidin stage of prophase 1 of meiosis. The next question, the complex formed by a pair of synapsed homologous chromosome is called. That is, when homologous chromosome come close together, they form a structure. This is also called the tetrastructure or bivalent structure. This structure is called the bivalent structure. This homologous chromosome come close together and they form a tetrad-like structure. They form a tetrad-like structure. Because of that, this is a tetrad-like structure. So this is a tetrad-like structure. This structure is also called bivalent. The complex formed by a pair of synapsed homologous chromosome is called bivalent or tetrad. The next question, during meiosis 1, the chromosome starts pairing. That means when the homologous chromosome come close together. In which phase of the prophase 1 of meiosis 1, the chromosome starts pairing. That means chromosome come close together. The answer is zygote. In zygote, the pairing homologous chromosome or synapsis takes place in the zygote stage of meiosis 1. The next question, during the metaphase stage of mitosis, spindle fibers attach to chromosome at. The question is very simple. During metaphase stage of mitosis, that is what happens in metaphase stage? In metaphase stage, all the chromosomes are aligned at the equator and chromosomes are connected to spindle fibers that is arising from the centriole to the kinetochore of the chromosome. So answer is kinetochore. A stage of mitosis is shown in the diagram. Which stage in it and what are its characteristics? From the diagram we need to answer the question. Look at the diagram. All the chromosomes lie at the equator. That means the chromosome that means the cell is in metaphase stage. And each of the chromosomes is attached to the spindle fibers arising from the centrioles, and these chromosomes are made up of two sister chromatids. So the answer is option B. The next question is, during gamete formation, the enzyme recombinase participates during gamete formation. That means in meiosis. Meiosis takes place in gamete formation. So when the enzyme recombinase participates, it is metaphase 1, anaphase 2, prophase 1 or prophase 2. The answer is prophase 1, because in prophase 1 there are some stages: leptodyne, zygote, pectide, diplotin and diakinesis. Okay, so where recombinase act because of the crossing over, the recombination of chromosome takes place, and in this stage recombinase become activated, at least to the occurrence of the meiosis 1. Hope you will enjoy these questions, and then if you like my video, kindly share the video and subscribe my channel. We will continue the same